Hey there, welcome to our second video for Endocrine for Biology 233. So we went over some basics last time about what hormones were, what target cells were, kind of some of the biochemical events that can occur with hormones, went over the main classes of hormones. But now we're going to talk about how those hormones can actually act on the cell. And there's two different ways that they can act on the cell. And one thing to think about before we get started is we mentioned that receptors can be inside the cell or they can be on the actual cell membrane. And so this is now a hint as to the two different ways that the cell can react to the hormone when it stimulates it. So let's switch over. This is where we left off last time. So we were at the bottom of page 10 of the lecture notes here, and we were talking about how hormones exert their effects on the target cells. And there are two systems here. So the first one is the second messenger system. And the second messenger system is the one that the amino acid based hormones use. So that's an important thing to note. This is used by water-soluble hormones that can be out in the blood plasma and can just go from the blood plasma and right onto a membrane-bound receptor. Okay? Now, the gene activation, and that's sometimes called direct gene activation. I'm actually going to add the word direct in, and the reason that I like adding direct in is because the hormone goes in to the cell and kind of interacts almost directly with the genes inside the nucleus. And so that's different than being on the outside and just interacting with the receptor. So in direct gene activation, we have lipid soluble, the ones that were steroid based or fat based, right? These are our second type of hormone, and they interact with intracellular receptors. We're going to go over each one of these next. So we're going to start with the second messenger system. Now, this picture is not in your book. There's a similar one, and it's on page 605. So why don't we at least write page 605, all right, there for you to look at. And I'm going to show it to you right now because I think I have it up. Yeah, here it is, page 605. This particular one is a diagram of kind of what happens. And they have some little guys there that are kind of acting as the different things, and they kind of show you. And it's completely a good system. They also have, if you have the online version, they have a video that goes over this. And it's probably a little more complex than you need, but for those who are visual learners, this might be a really nice way to actually kind of see what's happening there. Okay, so we will not watch that video right now, but instead what we're going to do is we're going to look at this same diagram here. Hold on one sec so I can make the page a little smaller, and we're going to look at it on our notes, and we're going to go through the notes and kind of annotate the notes and annotate the diagram. So the first thing I want to do is, really this diagram is two diagrams. And I'm going to draw right down the middle here, I'm going to draw a dotted line that goes all the way down the dotted line. Okay? Actually, it doesn't need to go that far. It can go right to there. Okay? And so on this side of this picture, we're going to stimulate the cell to do something. Okay? So we're going to tell the cell to do something, maybe increase the amount of production of something or how much it divides. We're going to make it divide more and go through cell division. On this side, we're showing a different hormone that would inhibit this cell from doing something. So if it was secreting something, this side of the picture is going to inhibit it from secreting. Okay? So there we go. Let's get it. So it's nice in the view of what we're looking at. All right, so the other thing you might notice on this screen is we have some numbers. 1, 2, 3, 4, 5. And there's numbers both for the stimulate and numbers for the inhibit. Now I'm going to just look at the stimulate one for a second, and I'm going to circle these so when we get down to the words below, we'll know where we're talking about. So there's the first, number 1. And number 2 goes and is this process here. Okay. Number 3 is this process right here. And then number 4 is kind of self-explanatory, and number 5 is kind of self-explanatory. So we are going to look at these 5 steps, and we're going to go through them. Those 5 steps are also listed in your book. If we go back to the book for a moment, I think it's up above here. Notice right here, 1, 2, 3, 4, 5. Those 5 steps are also listed in your notes, so you can read them in your book or in your notes. I'm going to focus on what we have in our notes. So, first thing is and remember, this is the second messenger system. This is the one. And the reason it's called that is because the hormone always stays outside, right? Here's the hormone right here, and it never goes inside. It stays out here. So we need other messengers to send the message from the outside to the inside. This would be like if you were walking up to a door, and maybe let's say the door had a window in it, and you went to the window and you knocked on the door and someone came up, and then you told them what to do, but you couldn't go inside. You could talk through the window and say, hey, would you go do this? And then they went and did what you wanted them to do. So you're like hormone A, you're staying out here. And what you're doing is you're somehow communicating to the stuff inside to do work for you. And that's what we're going to see right now. So the first thing that has to happen is the hormone has to physically bind to the target. And this is number 1. Okay, so number 1 there, we have to have the hormone bind to the target cell's receptor. And so if you look up here, you'll notice here's the hormone, hormone A, they're calling it. Here's the receptor, and we have it bind. What we're seeing here is things occurring over time. So first number 1 happens, then number 2 happens, then number 3 happens, then number 4 happens, then number 5. So this is like a little time lapse. So here it is without being attached. Here it is being attached, okay? So first thing, we interact the hormone and the receptor. This causes, when the hormone attaches to the receptor, it causes and allows a protein called a G protein, and specifically a GS protein, to bind to the receptor inside. So the hormone is causing this to happen, okay? So let's underline that. This interaction allows the receptor to bind to a GS protein. S stands for stimulatory. Okay? Stimulatory. This G protein is going to then become activated. And it becomes activated in step 2. And the way that we activate things is we can use energy. Oops, sorry about that. Put a 2 right there. Okay. And here, while I'm here, we're going to put a 3 right here. Okay. And we're going to put a 4 right here. And we're going to put a 5 right there. And then I won't have to come back and do that. So those are the 5 steps. Okay. So in step number 2, the G protein gets activated. And the way it gets activated is a GTP. Now, we've never talked about GTP. We've only talked about ATP. ATP is adenosine triphosphate. GTP is guanosine triphosphate. It is also an energy molecule. It's just a different kind. All right, so GTP gets used to and becomes GDP, just like ATP becomes ADP. And so the GTP will go and bind itself to the GS protein. Okay, so that's our next step. It will bind GTP and release a spent GDP. Now this guy is activated. So we have an activated GS protein, and it moves along the membrane and it goes to an enzyme. And this enzyme is called adenylate cyclase, and that's right here up at the top. If you look at the top of the page, adenylate cyclase. Okay, so we have our hormone A attaching with a receptor. We have that receptor allowing a stimulatory G protein to bind. We activate that G protein. Now the G protein moves along to an enzyme right here called adenylate cyclase. Adenylate cyclase's job is to convert ATP not into ADP, but actually go one step farther and go to cyclic AMP, so adenylene monophosphate. And so what that does is this cyclic AMP is now our second messenger. This is the thing that is going to tell the cell what to do. This is that other person on the other side of the window. Once you get their attention and you knock on the window and you speak and do all these things, those are all these first steps. 1, 2, 3, 4. And by the time you convey the message to the person, they are now the second messenger. And that's what cyclic AMP is. And notice that we can abbreviate it as CAMP. Okay? We can abbreviate it as CAMP. All right? Now, cyclic AMP then will activate things that are called protein kinases, and they do bunches of different things in the cell. So they basically activate a variety of physiological changes in the cell via these things called protein kinases, which do different work. And you can read up there in the box what kinds of things it might do. It might stimulate cellular secretion. It might open ion channels. It might do a bunch of things. Cyclic AMP gets degraded very quickly, all right? But due to cascade effects, there'll be millions of final product molecules. And the reason why is these protein kinases, the ACE at the end, means it's an enzyme. So they're going to act as enzymes and work very quickly and do lots of things. So even if one camp gets one protein kinase to do some work, that protein kinase might make thousands and thousands of final product molecules, whatever it's doing, whatever it's supposed to be, whatever its job is. So we get a lot of stuff. Now, one other note, and I'm going to put this in a different color so we kind of keep it separate from the original one. Some cell reactions even use a third messenger, and that third messenger is calcium. And it will bind to calmodulin, which is a receptor protein. And you could look in the book for details about this. But the main thing I want to get across is many of them just use a second messenger. Some will even, the cyclic AMP, will go and tell calcium to be a third messenger. So it's like the person inside then going to tell someone else, and then they're going to do the job. All right. Now, the thing that's kind of interesting, and I'll keep the color, why don't we put the color in as red here? We'll underline this in red, is we can reverse all this if an inhibitory G protein is stimulated. Okay? So the opposite of the above occurs if an inhibitory protein is stimulated. It shuts off the cell reaction. So you don't need to necessarily know how this works, but if you go up here to the top, look at hormone B. Hormone B is going to make an inhibitory G protein shut off the adenylate cyclase. So every step will be the same like it was over here, but instead of a stimulatory protein, we're going to put an inhibitory protein here, and this will shut off the process from happening. Okay? So that's kind of cool. One hormone can stimulate, another hormone can shut off. And we're going to see some of those hormone pairs later when we get to specific hormones and how they stimulate and inhibit cells to do work. Okay. So hopefully that makes sense. You can go back and look at it if you want. You can look in your book, watch that video. There's lots of ways that you can learn this. Now, I think direct gene activation is a little bit easier to understand, but we'll see what you think. There's less steps for one thing. I'm going to actually draw again on the thing here, and I'm going to put some numbers in, and I'm going to do it right now, and then we can come back and talk about them. Direct gene activation is on page 606. So first let's write that in, page 606. All right, so we have that, and then I want to put in the steps that we see there. So there's 1, 2, 3, and then I'm going to put in one more step way over here, 4. Okay, and then what I'm going to do is I'm going to come back to my picture. I'm going to zoom in a little bit, and I'm going to write those same numbers. 1, that's a 1. Here, let's make that look prettier. 1, and then we have a 2, and then we have a 3, and then we have way down here a 4. Okay, now let's go back up to the top and look at these together. We'll make this so we can see everything at one time. All right, so in direct gene activation, the hormone can go inside to the cell. Now, remember we said before, these are fat-soluble molecules. These are ones that come from steroids. So they are fat-soluble, so they don't float around in your blood plasma by themselves. They're usually delivered to the cell on a carrier protein. All right? And this is because they're fat-soluble. If they weren't, they might all glob together and form a little fat droplet, and we could get a embolus situation. So we don't want that. So the steroid hormone is delivered to the cell via protein, and then the steroid hormone will diffuse through the plasma membrane, and that's what happens in step 1 here. It's going to diffuse through the plasma membrane of the target cell. And you can see that where the 1 is, it goes right in. And it actually goes all the way inside to the nucleus. This purple that you see there, right here, this purple, this whole purple area, is the nucleus. And so it goes through a nuclear pore, and it goes inside the nucleus. Once it's inside, it binds to this thing. There's different names for it. We call it a receptor chaperonin complex. I think the book just says receptor hormone complex. I kind of like the chaperonin idea, because what happens if you're looking here where number 2 is, if you're right here, okay, you see that there is a molecule that is attached to the receptor. And the steroid hormone and the molecule come together. So this chaperone molecule kind of brings them together. So think of like a chaperone taking someone to like a dance, like your father taking you as your chaperone to a dance. But then he doesn't stay and dance with you. He lets someone else dance with you. And so then the couple forms, and the chaperone goes away, maybe goes out to the car and waits till the dance is over, goes to the side of the dance stage, dance floor. So the second thing that will happen is we will have the hormone binding to an intracellular receptor chaperonin complex. Then the chaperone detaches, and then we just have the hormone receptor complex. Okay? The third thing that then happens is this hormone receptor complex, which is right here, will move down and go directly to DNA. This is why it's called direct gene activation. Okay, it goes into the nucleus, finds the DNA, and it binds to a receptor on the DNA. And it causes the production of mRNA. Now, if you remember what mRNA does, mRNA goes through transcription, remember this from intro bio, and goes through translation. And when that happens, we get a new protein made. So this hormone goes all the way in, and it will produce specific protein molecules and activate the cell. Right? So steroid molecules go this way. They go into the cell and directly activate genes to produce proteins, and then those proteins do things for the body. So one way that is good is to compare and contrast the direct gene activation with the second messenger system. So think about that for a minute. First, the second messenger system uses amino acid-based ones. The hormone always stays on the outside. The hormone uses a second messenger or sometimes a third messenger to get the cell activation to occur. While on the other hand, the direct gene activation, the steroid goes inside the cell. It combines with the receptor, and then it goes from the combined steroid and receptor go down and attach directly to DNA and cause transcription and translation. And that's how we make protein, or we activate the cell. So there's a lot of differences between them. Not only the type of hormone, but the direct way or indirect way that the messages get sent into the cell to activate the cell. So review that and see if all that makes sense. You can, again, look in your book. There's other, you can go online and do a little research on those if you want. You can type in second messenger system and find, videos about it if what I'm saying is confusing to you. All right, so we're going to talk a little bit more here before we wrap this one up. And what we're going to do is we're going to just do this page here, I think, which is the control of hormone release. Most endocrine glands use a negative feedback system to release their hormones, to make and release hormones. That's probably not surprising to you because from intro bio and from 231, we tend to use a lot of negative feedback systems. We're going to actually only talk later about one hormone that uses positive feedback. All the other hormones that we use this entire time that we talk in the next week or two are going to be negative feedback. So that's interesting. But there are different ways that we can stimulate the release of hormones. And this is what we want to talk about. And there's three main ways, and we'll highlight these, all right, and then talk about some examples and specifics. And these are all on page 607. Let's zoom in here and write that right there. Okay, so on page 607 is this picture, all these pictures here in your book. And the book does a really good job, actually, and I might not fill in these just so it'll be easier, and maybe I'll go right to the book. So first, let me flick over here to the book. Oh, it wants me to go away. It thinks I should leave, but we're not ready to leave yet. Okay, and by the way, there's direct gene activation on page 606. You can look at all the steps. They have 5 steps. I only had 4, but it works about the same. And then once we get past that, we're going to talk about the types of release. Okay, so there are 3 types of release, and they're all right here. And notice this picture is the same as the picture in your notes. It's just one is horizontal and one is vertical. And I think what we'll do is we'll look at this a little bit, but let's first highlight your notes, then we'll come back here, okay? So if we go back to our notes, there are 3 ways that we can stimulate endocrine glands to release hormones, okay? So these are the 3, whoops, let's erase that. We can release the hormones in 3 ways, okay? First one, I always want to change everything here. Okay, the first one is humoral, right? Humoral means the humors or the fluids of our body. And so what this is going to look at is it's going to look at specific levels of ions and nutrients in the blood. And that's what's going to control the release of the hormone, okay? So think of a couple things that you might already know that might do that. Glucose levels. If your blood sugar is high or low, we might release hormones like insulin and glucagon. Calcium levels. If calcium's high or low, we often will uptake extra calcium in our bones, or if it's low, we'll release calcium from our bones. So PTH and calcitonin do those things. Sodium. We know that sodium works with aldosterone and also in opposition ANP. So the amount of sodium in our fluid sometimes determines whether we're going to increase or decrease our blood pressure. So one example of that one is calcium levels, and that's right here. And I'll talk about that in just a minute on in the book, because the book has it all filled in, and I'm not going to fill that in there. I'll just make a big mess of it. Now, neural is next. So neural ones, what do we do with neural ones? These are exactly what it says. Nerve impulses stimulate the release of hormones. Now, you've already looked at one of these. This example over here might look kind of familiar to you from 231. Do you remember when sympathetic fibers stimulated the adrenal medulla, causing epinephrine and norepinephrine to be released? That was our exception that we talked about, where it never synapsed. That's an example of neural. There's some others too, like oxytocin and ADH are also released neurally. We'll talk about those later. This example right here, next to the green here, is this sympathetic fiber one. So we'll go back to that in just a minute. The third way that we can release hormones is hormonally. Okay? Hormonally. And it's well named because it just means that a hormone will cause the release of a hormone. And this happens a lot. Hormones will stimulate glands to release other hormones. The hypothalamus releases a lot of hormones that are called releasing or inhibiting hormones. And they either turn on and release more hormones, or they inhibit and don't tell glands to stop releasing hormones. And that's what we're seeing here. Okay, let's go back over to the book for a second and look at this page. So this page number, as we said, is page 607. And we're going to flip back over for a minute. So let's look at the first one, humoral stimuli. So this one is altered by levels of certain ions and nutrients. So the example we're giving here is low blood calcium. If we don't have a lot of blood calcium, the parathyroid gland will release a hormone, PTH. And PTH will go through a couple different steps, which we'll talk about later, to increase calcium in our body. So a certain level is off, it releases a hormone. That low level causes a hormone to be released, and that hormone then will try to solve the problem and bring us back into homeostatic balance. Neurally, so here's our exception. Remember, we had a preganglionic fiber go all the way, never synapse, go into the medulla of the adrenal gland and release epinephrine and norepinephrine, adrenaline, into our bloodstream. So that is direct neural stimulation. And that's what we see there, neural stimulus. The third one, hormonal stimulus. So one hormone's released, and that released hormone goes to a gland and tells that gland to do something. So the example here is there are releasing hormones that come from the hypothalamus. They go out into the pituitary gland, and the pituitary gland sends hormones that go stimulate glands, and then those glands release hormones. So we have hormones releasing hormones, releasing hormones. So all of these arrows are, this arrow here is a hormonal stimulus. So take a look at those, make sure you're understanding that stuff. I think we're going to stop there, and we're going to pick up on the very next page of our notes. Let's go to where that is so you'll be ready next time. And we're going to start talking about the actual endocrine organs, and we're going to go through each organ in order and what hormones it produces. We're going to start with the pituitary gland and kind of work our way throughout the body. All right? So thanks so much for watching and listening. If you have any questions, ask your instructor via email or through Moodle, and either I or your other instructor will help you out as best we can. All right? All right, have a great day. Thanks.